Press 2 fingers to the base of your own spine. That small, blunt point is not just a bone, it is the end of a tail. Every human embryo grows one before the body quietly dissolves it. The code for building it was never deleted. It is still folded inside every human cell, switched off, while most primates keep that switch on for their entire lives. So why do human genes still carry the code for a tail? Picture the body 4 weeks after conception, long before it has a face worth naming, folded into itself in total darkness, smaller than a grain of rice, and at the base of what will become its spine, something extends outward that no textbook diagram of a finished human ever shows. It is not a swelling. It is not a fold of skin waiting to become something else. It is a true tail, built from actual vertebrae, jointed and structured the same way the rest of the spine is being built at that very moment, growing outward as a distinct, countable extension of the body's central column. For a short stretch of days, this tail is not a small, hidden detail tucked away at the end of development. Relative to the rest of the embryo's length, it is one of the more noticeable features taking shape, curving forward along the body the way it does in the early embryos of many other vertebrates, because at this stage, the plan being followed is shared far more broadly across the animal world than most people ever get told. Nobody explains this to a person growing up. Nobody mentions, in the years spent learning about the spine, the ribs, the skull, that the same body, now hearing about it once, carried a genuine, structural tail, with its own bones, its own blood supply, its own thread of nerve tissue running through it, doing exactly what a tail is built to do at that stage, existing for a reason that has nothing to do with decoration. At its most developed point, before any dismantling begins, this tail carries more segments of spine than will ever remain visible in the finished body, a brief surplus of vertebrae built and then later folded away, present just long enough to do whatever quiet structural work it is doing at that stage of construction, before the body moves on to other priorities entirely. There is no dramatic image of this moment hanging on a classroom wall, no diagram passed around in an introductory biology class with an arrow pointing to it and a label announcing what it is. It exists mostly in specialized textbooks, in the plates used to train people who will spend their careers studying exactly this kind of thing. Images of a curled, translucent form no larger than the last joint of a finger, with a small tapering extension trailing from what will become the lower back. Researchers studying these early weeks of human development can use the length and stage of that extension almost like a clock, because it appears and recedes with enough consistency that its presence or absence helps place an embryo at a very narrow window of days. That level of precision is not the behavior of a random leftover fragment. It is the behavior of something following a schedule closely enough that its stages can be read like one. Then, within a matter of days, something begins to reverse. Around the 8th week, the tail that had been extending outward starts disappearing, not through injury, not through some outside force tearing it away, but through a process built into the cells themselves. The technical name for it is apoptosis, and it is worth sitting with what that word actually means, because it is easy to hear it and picture something violent, something going wrong. It is closer to the opposite. Apoptosis is not damage. It is not decay. It is an internal, choreographed signal that tells specific cells, on a specific schedule, to fold themselves down and cease in an order precise enough that the surrounding tissue is left clean, without scar, without gap, without any outward sign that something was ever there. The cells that made up the tail do not die because something attacked them. They die because they were always going to, on a timetable written into them before they ever formed. To understand why this reads as controlled rather than chaotic, it helps to compare it with what happens when tissue is damaged by an actual injury. An injury leaves debris. It leaves inflammation, swelling, a visible scramble from the surrounding area as the body responds to something it did not plan for. None of that happens here. The cells that made up the tail do not rupture outward and leave a mess for the rest of the body to clean up. They shrink inward, methodically, breaking their own internal material down into small, manageable pieces that neighboring cells then quietly absorb, the way a controlled demolition leaves a clear site rather than a scattered pile of debris. This is the same basic family of process, in a different context, that carves separate fingers out of a hand that starts out with no visible gaps between them, dissolving away the webbing that once connected them one by one, quietly, without any of the drama the word disappearance tends to imply. The disappearance of the tail belongs to that same quiet category of events. The body's willingness to build something, use it as scaffolding for what happens next, and then remove exactly what is no longer required without disturbing anything around it. Every single human being who has ever lived passed through this same stage in this same order, on a timeline consistent enough that embryologists can look at an unlabeled embryo of the right age and know, without guessing, whether its tail should still be present or should already be gone. Almost nobody's body is exempt from this pattern, and even the rare departures from it end up confirming the rule rather than breaking it. Right now, at the base of the spine of the person listening to this, there is a small triangular arrangement of fused bone that is not a separate design choice, not something added on afterward, not a coincidence of shape. It is the closed end of a structure that used to extend further, present at some point in every human who has ever existed, hidden beneath skin that gives no outward hint of what used to be there. What makes this strange is not that the tail disappears. Structures disappear during development constantly, and most of the time nobody would think twice about that. What makes it strange is the order of events. The body does not simply skip building a tail and move directly to the version without one. It builds the tail first, complete with its own vertebral segments, its own connective tissue, its own place in the body's early architecture, and only after committing real biological resources to constructing it does it turn around and take apart what it just made. If the final destination was always going to be a body without an external tail, the more economical path would seem to be never building one at all. Skip the step. Save the material, the signaling, the energy an embryo has to spend, laying down bone and tissue that will only be dismantled again within a matter of weeks. And yet that is not what happens, not in this body, not in any human body, not once across the entire span of a species that has never produced a single member who wasn't born having passed through exactly this same detour. Evolution is often described as efficient, as a slow process that trims away anything unnecessary given enough time and enough generations. And yet here is a structure that every human embryo builds without exception, carried for weeks, supplied with its own share of the embryo's limited early resources, only to be dismantled before it ever performs the one function most people would guess a tail exists to serve. If the story usually told about the body is one of things earned and kept because they are useful, this sits closer to a counterexample, something invested in and then subtracted inside a process that supposedly favors saving effort wherever it can find a way to. This is not a flaw being corrected partway through construction. A flaw would be inconsistent. It would show up differently from one person to the next, would look like an accident when compared across enough bodies. This looks like the opposite of an accident. It happens the same way at the same stage, with the same clean outcome, in case after case, which is closer to the signature of a plan being carried out than a mistake being cleaned up after the fact. Something in the deep architecture of human development insists on building this structure before removing it, as though the removal itself somehow depends on the structure having existed first, as though there is no shortcut available that arrives at the same tailless outcome without passing through the tailed one on the way there. Nobody carrying that small blunt bone at the base of their spine chose this sequence, and nobody alive today was consulted about whether building first and dismantling second was worth the trouble. It happened before there was a nervous system complex enough to be aware of anything happening at all, decided somewhere far upstream of conscious life, in a layer of instruction that runs quietly underneath a body, long before that body has any say in what gets built inside it. Older than memory, older than language, older than anything the person carrying it could ever consciously recall. And that is where the strangeness settles, at the base of the spine, in a bone too small to draw attention to itself and too permanent to ever fully disappear. If building it was somehow required before it could be safely taken away, then the smooth, fused point under the fingers of anyone listening right now is not simply a leftover fragment of something no longer needed. It is the visible signature of a decision made deep inside the code of the body, one that chose construction and demolition over never starting at all, for reasons that have not been explained yet, and will not be explained for a while still. Somewhere in a forest canopy right now, a monkey shifts its weight along a branch, and the tail trailing behind its body is not a passive decoration hanging off the spine. It tightens, curls, presses down against the bark for an instant of extra grip, doing real mechanical work the same way an arm or a leg does, correcting balance before a shift in weight becomes a fall. Watch closely enough, and the tail seems almost to think for itself, adjusting a fraction of a second before the rest of the body commits to a jump, testing the next branch's give before any paw or hand makes contact with it. In some species, that tail does even more than balance. Certain monkeys in the forests of Central and South America can wrap the tip of their tail around a branch with enough strength to hang their entire body weight from it, freeing both hands to reach for fruit, to groom, to carry an infant, using the tail less like an accessory and more like a fifth limb built for grasping. Some of these tails even carry their own fingerprint-like ridged skin at the very tip, a texture normally associated with hands, evolved there for exactly the same reason it exists on a palm, to improve grip on a surface that will not forgive a loose hold. For those animals, the tail is not a leftover of anything. It is active, load-bearing, essential to how they move through the exact three-dimensional world they live in every single day of their lives, without exception. Now bring the base of a human spine back into the picture. Whatever remains there, sealed and fused beneath the skin, sits in stark contrast to what is happening in that canopy. And the contrast gets sharper the closer the comparison gets, because monkeys are not some distant, unrelated branch of the animal world. Many of them belong to the same broad order primates do, sharing a body plan, a skeletal layout, a nervous system built along the same basic blueprint as the one reading or hearing about it right now. If a tail is such a functional, load-bearing piece of equipment for close biological relatives living in trees, its total absence starts to look less like an obvious inevitability and more like something that needs an explanation. Here is where the picture widens further, past what most people expect. The absence of an external tail is not a trait unique to humans among the primates. Every great ape shares it. Chimpanzees, the closest living relatives humans have no tail. Gorillas, heavier and more ground-bound, have no tail. Orangutans, spending nearly their entire lives moving through trees, have no tail. Gibbons, some of the most acrobatic tree-dwelling primates alive, swinging hand over hand through the canopy at speeds few other mammals can match, also have no tail. Draw a line around every species classified as an ape rather than a monkey, and not one of them carries an external tail into adulthood. This is not a coincidence, scattered randomly across unrelated lineages. It is a shared trait, present across an entire branch of the primate family tree, distinguishing that branch cleanly from the tailed monkeys sitting right next to it on the same evolutionary map. None of these animals move through their world the way a tailed monkey does, either. A gibbon crossing a gap between two trees does not correct its balance with a tail, because it has none to use. Instead, its entire body relies on enormously long arms, swinging hand over hand in a motion built almost entirely around the shoulders, using momentum and grip strength in place of whatever balancing work a tail might otherwise be doing mid-air. A chimpanzee or a gorilla moving along the ground shifts weight through its knuckles, using the front limbs almost like a second pair of feet, a style of walking found nowhere among the tailed monkeys watched a moment ago, balancing along branches. Something about the way these animals move, the way weight travels through their bodies, developed around the absence of a tail rather than trying to replace it with a hidden substitute. The tail was not swapped for some quieter equivalent. It was removed, and everything downstream of that removal, every joint, every muscle, every habit of movement, developed as though a tail had never been part of the plan at all. Compare a skeleton from either group side by side, and the difference is not subtle. Where a tailed monkey's spine trails off into a long, flexible extension of small bones, an ape's spine simply stops, closing off cleanly at a short, blunt point, as final and deliberate looking as a sentence ending in a period rather than trailing into nothing. That detail changes the shape of the question completely. A person running a finger along the base of their own spine might reasonably assume, without ever being told otherwise, that this is simply what a human is, that the absence of a tail is a fact belonging only to this one species, set apart from the rest of the animal world by its own specific design. But that assumption does not survive contact with the wider primate family. Whatever happened to remove the tail did not happen to humans specifically. It happened earlier, and it happened once, to a shared ancestor old enough that its descendants would eventually branch out into every living ape species that exists today, humans included. The trait was not built into the human body as something unique. It was inherited, passed down the same way eye color or blood type gets passed down, arriving already decided by the time any human embryo starts forming its own brief, temporary tail only to dissolve it weeks later. Whoever is listening to this right now, resting somewhere quiet, carries the exact same inherited outcome as a chimpanzee resting somewhere in a forest, thousands of kilometers away, the same small fused bone doing the same quiet job at the base of the spine, shaped by the same decision made in the same distant past, long before either species had a name for itself. There is something almost humbling in that. The hook that opened this journey framed a very personal question, pointed at one specific body and one specific bone. It is tempting to hear a detail like that and assume it says something distinctive about being human, one more entry on the list of traits that supposedly separate humans from the rest of the animal kingdom. But the moment the comparison widens to include chimpanzees, gorillas, orangutans, and gibbons, that assumption collapses. This is not a marker of human uniqueness. It is a marker of shared ancestry, evidence of a decision made so far back in time that every one of humanity's closest living relatives inherited the exact same outcome. The bone at the base of a human spine and the equivalent structure at the base of a chimpanzee's spine trace back to the same missing feature, removed from the same shared blueprint, long before either species existed as something separate from the other. A person could spend a lifetime believing their body was written from a uniquely human draft, only to find, at the smallest and least discussed bone in their entire skeleton, that this particular page was copied, unchanged, from a much older manuscript shared by an entire family of species, which raises a harder question than the one the hook opened with. If the absence of a tail is not a human trait at all, but an ape trait, inherited rather than independently evolved, then something must have happened to the ancestor all of these species share, something that made carrying a tail stop being worth the cost, in a way strong enough to affect every single lineage that descended from it afterward. Monkeys living today, close relatives on the same family tree, kept their tails and kept using them for exactly the kind of active, functional work described at the very start, swinging, balancing, gripping branches with the tip. The ape lineage did not follow that same path. Something diverged. Something about the ancestor at the root of every tailless ape alive today stopped needing what a tail provides, badly enough that the trait disappeared and stayed gone across every branch that followed, without a single documented reversal in millions of years of descendants. No answer has been offered yet for what that something was. The comparison so far only establishes the shape of the mystery more precisely than the hook alone could. This is not a question about one body. It is a question about an entire branch of the primate family tree, spanning species separated today by continents, habitats, diets, and ways of moving through the world, all of them united by the same missing feature, all of them carrying some version of that same small fused bone where an active, working tail used to be, somewhere back along that shared line, in an environment that has not yet been named, under pressures that have not yet been described. An ancestor common to humans and every living ape stopped being served by the very structure that still serves monkeys well today. There is no fossil skeleton anyone can point to yet in this account, no date, no forest, no clue about what that ancestor's daily life even looked like at the moment its own tail started shrinking. All that has been established, for now, is the size and shape of the family this happened to, wide enough to include every ape alive on the planet today, humans among them, and old enough that no single living species can claim ownership of the moment it happened. What that environment looked like, and what exactly made a tail worth losing, is still sitting, untouched, waiting. Step back now, far enough that the chimpanzee and the monkey from a moment ago are no longer separate animals living in the same modern forest, but the same population, standing in the same place, at the same point in time, not yet split into two different futures. That moment sits somewhere between 20 and 25 million years in the past, long before there were continents shaped the way they are now, long before grasslands had spread the way they eventually would, in a world still thick with forest across huge stretches of what is now Africa and parts of Eurasia. Warm, wet, and largely covered in trees, this was a climate that rewarded a body built for moving through branches rather than across open ground. Air thick with humidity, canopy layered so densely in places that daylight would have reached the forest floor only in scattered fragments, filtered through leaves that had never once been touched by a human hand or looked at by a human eye. Somewhere inside that unbroken green world, among countless other primate populations living out their own separate lives, one particular lineage was quietly approaching a fork in its own story that its members had no way of noticing while it was happening. This is not a guess pulled from imagination. The timing of that fork can be pinned down with real evidence, arrived at from two completely different directions that happen to agree with each other. One direction comes from the ground itself, from fossilized bone recovered from rock layers old enough to date reliably, pulled out of deposits in East Africa that preserve fragments of early primates, already showing traits associated with the ape body plan rather than the monkey one. The other direction comes from something far less visible than a fossil, sealed inside the cells of every living monkey and ape alive today. Every time a genome is copied from one generation to the next, small errors creep in, tiny changes to the genetic code that accumulate slowly and steadily across enormous stretches of time. Two species that split from a shared ancestor a long time ago will have had more generations to accumulate these small differences independently than two species that split apart more recently, which means the amount of genetic difference between any two living species can be used almost like a clock, ticking at a roughly steady calibratable rate. By comparing how much the genetic code of different species has drifted apart over time, and by using a rate of accumulated change that has been calibrated against fossils whose ages are already known, through other independent methods, researchers can estimate how long ago two living lineages shared a single common ancestor. When the fossil evidence from the rock and the genetic evidence from living bodies are lined up next to each other, they point to the same narrow window, somewhere close to that 20 to 25 million year mark, as the moment the branch leading to every living ape split away from the branch leading to today's tailed old world monkeys. There is something worth sitting with in the fact that two such different kinds of evidence, one dug out of stone and one read out of a living genome, converge on the same number without either one depending on the other. It means the timing is not resting on a single fragile source that could later be overturned by one new discovery. It is a conclusion two very different scientific methods keep arriving at independently, which is generally the strongest kind of evidence available for a question this old. Neither method can say precisely which individual animal in that ancient population carried the first version of whatever change eventually mattered. What both methods agree on is the window during which the population as a whole was clearly dividing into two futures, one that would go on to produce every tailed monkey species alive today, and one that would go on to produce every tailless ape alive today, humans included among the second group, a skeptic could reasonably distrust either method taken alone. Fossils are rare, fragmentary, and only ever capture the smallest sliver of everything that once lived. Genetic clocks depend on assumptions about mutation rates that can vary between lineages and need careful calibration to trust. But it is far harder to dismiss two independent, differently flawed methods when they keep landing on the same narrow window rather than drifting apart, which is exactly what happens here. It helps to say plainly what this window of time actually means, measured against anything more familiar. 20 to 25 million years is a span roughly 4 times longer than the entire existence of the genus that modern humans belong to, and many times longer than the existence of anything resembling a human ancestor walking upright on 2 legs. There were no stone tools yet, no fire, no hominins of any kind recognizable as belonging to the human family, not even close relatives distant enough to be called ancestors in any meaningful sense. Whatever happened to remove the tail from this population happened in a world with no humans in it at all. No early humans, no proto-humans, nothing occupying that particular branch of the primate family tree yet. It happened to a population of forest-dwelling primates going about lives shaped entirely by pressures that had nothing to do with humanity, because humanity, in any recognizable form, would not exist for tens of millions of years afterward. And yet, the small, fused bone sitting right now at the base of the spine of whoever is listening to this is a direct, unbroken continuation of that same ancient population's story, carried forward through an unimaginable number of individual lives, generation folding into generation, all the way from a warm, humid forest that no human eye ever saw, to this exact moment, this exact body, resting somewhere quiet tonight. That timing changes how the question has to be asked. It is easy, hearing about tail loss for the first time, to imagine some later human ancestor slowly losing a tail as part of becoming more human, as though the tail were shed somewhere along a path leading specifically toward the species listening to this now. That picture gets the order of events backwards. The tail was already gone from the shared ancestral line tens of millions of years before anything resembling a human branch existed to walk away from. Humans did not lose a tail on the way to becoming human. Humans simply arrived, many millions of years later, into a lineage that had already been tailless for an almost incomprehensible stretch of time, inheriting an absence that had been settled long before there was anyone or anything called human to inherit it. Picture that ancient population once more, moving through a canopy so continuous that the forest floor beneath it barely mattered, climbing, reaching, shifting weight from branch to branch, the same way the tailed monkeys from a moment ago still do today. Somewhere among those individuals, in ways no fossil can capture directly, something about daily life in that environment was quietly making a tail less valuable than it had been for their own ancestors, and more valuable, presumably, for the branch that would eventually become today's monkeys, since that branch kept its tail while this one did not. No one alive to witness it would have called it a turning point. It would have looked, day to day, like nothing more than ordinary life, ordinary feeding, ordinary movement, through an ordinary forest, the kind of unremarkable repetition that never announces itself as history while it is happening. Both branches were living through the same broad slice of geological time, likely in overlapping or neighboring forests, facing many of the same predators, the same fruiting trees, the same basic three-dimensional problem of getting a body safely from one branch to another. Yet one branch's answer to that problem increasingly did not seem to require a tail, while the other branch's answer still did. Nothing about this moment was chosen the way a person chooses anything. No individual animal decided to stop needing a tail, any more than any individual animal decides today which traits get passed to the next generation. Something about the pressures of that particular time and place simply made carrying a full, functional tail less advantageous for one lineage's specific circumstances, generation after generation, until whatever the underlying cause turned out to be had shifted the whole branch permanently. There is no single dramatic event to point to here, no sudden catastrophe, no one storm or one predator or one collapsing tree that could explain a change this large. Change on this scale, spread across a population over a span of time too long for any individual life to register, tends to come from something quieter and more constant, some steady feature of daily existence repeated so many times, across so many generations, that its effect eventually becomes impossible to reverse. What exactly that underlying cause might have been, what was actually happening in the daily lives of these forest-dwelling primates that made a tail worth less to them than it remained to their tailed relatives, is a separate question from the timing itself, one the timing alone cannot answer. The clock has been set. The window has been named. 20 to 25 million years. A warm and heavily forested world. A single population standing at the edge of two different futures, one that would keep its tail and one that would not. What was actually happening inside that window, in the branches and canopies of a world that existed tens of millions of years before anything human, in the daily habits and movements of animals that never once considered themselves the ancestors of anything, is still waiting to be looked at directly. Watch a tailed monkey cross a branch, and the body moves the way a person might cross a narrow beam, with arms out for balance, walking along the top of the support, four limbs planted on the same surface, weight constantly shifting from side to side with every step. In that posture, a tail trailing out behind the body becomes something close to a counterweight, swinging and adjusting in the opposite direction of whatever the torso is doing, the same basic physics a tightrope walker relies on when holding out a long pole to keep from tipping. Take that counterweight away and the whole system becomes noticeably less stable, which is part of why tailed monkeys rarely seem to hesitate before committing to a jump between branches that would look, to an untrained eye, dangerously far apart. Now picture a different way of moving through the exact same forest. Instead of walking along the top of a branch, an animal hangs beneath it, gripping with its hands, letting gravity pull the body straight down rather than sideways off balance. Moving this way, the challenge is no longer staying upright on top of a narrow support. It becomes a matter of swinging from one handhold to the next, using momentum built from the arms and shoulders, the body hanging below whatever it is holding onto rather than perched above it. This style of movement, common among the great apes and taken to its most extreme form by animals that swing hand over hand at speed through dense canopy, changes the entire physics of the problem. A body hanging below a branch is already being stabilized by gravity itself, pulled straight down into a natural, low center position, in a way that a body balanced on top of a branch never is. Whatever counterbalancing work a tail does for the tightrope walker's version of moving through trees, the hanging version simply does not need in the same way, because the body's own weight, pulled downward, is doing a version of that stabilizing job on its own. This is the leading explanation offered for why a tail might have stopped being worth keeping for the branch of the primate family that eventually gave rise to every living ape. If an ancestral population was shifting, generation after generation, away from walking on top of branches and toward hanging and swinging below them, the tail's original job would have mattered less and less with each passing generation, until a body built without one performed just as well, or perhaps even better, freed from carrying and controlling an appendage that had stopped pulling its weight. Carrying a tail is not free in any biological sense. It requires its own bone, its own muscle, its own blood supply, its own share of nerve tissue devoted to controlling every subtle adjustment it makes, resources a growing body has to produce, and a moving body has to power with every single use. A structure that once justified all of that cost by preventing falls during careful, top-of-the-branch travel would, under this explanation, have slowly stopped earning its keep as more and more of a population's daily movement shifted toward a style of travel that did not depend on it in the same way. It is worth pausing here to notice what this hypothesis actually claims about the small fused bone sitting at the base of the spine of whoever is listening to this right now. It is not simply a mistake or a random accident of development. If this explanation holds even partly true, that bone is the residue of an old physics problem that an ancestor's body eventually stopped needing to solve. A structure quietly retired the moment gravity itself started doing enough of the stabilizing work that the original solution became unnecessary weight to carry. Some researchers extend this idea even further, connecting it to what happened much later, long after the tail was already gone, when certain branches of this now tailless lineage eventually came down out of the trees altogether and began moving upright on two legs. The idea is that a body already adapted to hanging with its spine oriented differently than a horizontal, branch-walking monkey's spine, already carrying its weight in a more vertical alignment, may have been sitting closer to the starting position an upright, two-legged posture would eventually require. Heavier ancestral apes moving increasingly along the ground would have relied even less on a tail for balance, their weight now carried through four sturdy limbs planted directly on solid earth rather than distributed across branches high above it. Another layer supporting the same basic idea that a tail's usefulness was quietly draining away the further a lineage moved from balancing atop thin branches. That connection remains a proposal rather than a settled fact, one thread among several that researchers are still weighing against each other, but it gives the locomotion explanation a reach that extends well past the original disappearance of the tail itself. There is a felt logic to all of this that makes it easy to accept as a finished answer, and this is exactly the point at which it becomes worth resisting that pull. Somewhere in a forest right now, a small, extraordinarily agile ape spends nearly the entirety of its life in the canopy, rarely if ever descending to solid ground, moving between trees, not by walking along branches the way a tailed monkey does, but by swinging hand over hand at remarkable speed, covering long gaps between supports through nothing but momentum and grip, sometimes releasing both hands entirely mid-swing, briefly airborne with nothing holding the body to anything at all before the next branch arrives. This is the gibbon, and by every measure that matters for the locomotion explanation, it should be the clearest possible exception to the rule. It never left the trees. It never adopted a ground-dwelling, upright lifestyle the way the explanation's extended version imagines happening later in other branches of the ape family. If tail loss was simply about no longer needing to balance on top of branches because life had moved to the ground, an animal that spent its entire existence swinging through canopy, having never once given up an arboreal life, would be the one ape most likely to have kept some version of a tail, or to have regrown one if an ancestor briefly lost it and then returned to a lifestyle that still seemed to call for it. That is not what happened. The gibbon has no tail at all, the same clean fused absence found at the base of every other ape's spine, despite living a life that never stopped requiring exactly the kind of arboreal agility a tail is supposed to help with. This detail does not disprove the locomotion explanation outright. It complicates it in a way that is worth sitting with, rather than rushing past. A body hanging beneath a branch, swinging at speed between handholds spaced sometimes many body lengths apart, still has to manage momentum, still has to control rotation through the air during the moment between letting go of one branch and catching the next, still has to avoid the kind of uncontrolled spin that would make catching the next handhold nearly impossible. It is not obvious, examined closely, that this kind of high-speed aerial travel needs a tail any less than careful, deliberate walking along the top of a branch does. And yet the gibbon manages all of it without one, using instead an entirely different set of adaptations, extraordinarily long arms, unusually mobile shoulder joints, a light and compact body built for exactly this kind of travel, none of which include a tail doing any part of the stabilizing work. What this leaves is a genuine, unresolved tension rather than a tidy conclusion. The locomotion explanation captures something real about the physical difference between hanging below a branch and balancing on top of one, and it fits comfortably with what is known about how most tailed monkeys move compared to most apes. But it does not fully account for an animal that kept the arboreal lifestyle the tail was supposedly no longer needed for, and lost the tail anyway. Something else may have been happening alongside the shift in movement style, some other pressure operating on the shared ancestor of every ape, Gibbons included, that made losing the tail worthwhile regardless of exactly how each descendant branch eventually chose to move through its own particular environment. Perhaps the earliest version of the change happened only once, in that single shared ancestor described earlier, for reasons that had nothing directly to do with any one descendant's later lifestyle, and every branch since, Gibbons included, simply inherited the outcome without needing a locomotion story of its own to justify it. Perhaps the true cause sits somewhere underneath all of these movement patterns entirely, in the body's own machinery rather than in the physics of any particular way of crossing a forest. The question of why has not been answered cleanly here, and it may not be, not entirely, from this angle alone. What can be answered with far more certainty than any environmental story can currently offer is a different question entirely. Not why a tail stopped being worth keeping, but how a body built to grow one, in every single embryo, generation after generation without exception, ever managed to stop finishing the job cleanly enough that the change could spread through an entire lineage and hold for tens of millions of years without a single confirmed reversal, gibbon and human included alike. Every structure discussed so far, the tail bud rising and dissolving in a 4-week-old embryo, the fused bone at the base of an adult spine, the ancient population splitting into tailed and tailless futures 20 million years ago, all of it traces back to instructions written inside a single gene, one specific stretch of DNA, carrying a name most people will never hear in their lives. Geneticists call it TBXT, though for much of the 20th century it went by an older name, brachyury, drawn from Greek roots, meaning short tail, given to it nearly 100 years ago after a researcher studying laboratory mice noticed something odd running through certain litters. Some of the pups were being born with tails noticeably shorter than their littermates, or with no visible tail at all, and breeding experiments traced the difference back to a single, altered gene passed down through specific family lines. Long before anyone understood what that gene actually did inside a developing embryo, its effect on tail length alone was distinctive enough to earn it a name built entirely around that one visible trait. That naming turned out to be almost too narrow for what the gene actually does, because tail length is only the most visible, most easily noticed part of a much larger job this single gene is responsible for. To understand why, it helps to go back further than the tail bud discussed earlier, back past the 4th week of development to something happening only days after conception, before the embryo has anything resembling a recognizable shape at all, still just a flat, layered disc of cells no larger than the head of a pin. At this stage, a small cluster of cells is undergoing one of the most important reorganizations a body will ever go through, sorting itself into distinct layers that will eventually become every major system a person has. Skin and nerve tissue from one layer, internal organs from another, and muscle, bone, and connective tissue from a third layer called mesoderm. TBXT sits near the center of the signaling that makes this sorting happen correctly, active during a stage of development called gastrulation, widely considered by embryologists to be one of the single most consequential events in the formation of any vertebrate body. The moment a formless cluster of cells first commits to becoming something with a front, a back, a top, and a bottom. It also plays a critical role in laying down a structure called the notochord, a temporary rod of tissue that runs along the length of the very early embryo and acts as a kind of scaffold, an early version of a body axis that the rest of the developing spine will eventually form around. None of this has anything to do with a tail yet. This is happening days before a tail bud exists, in a process responsible for giving the whole body its basic front-to-back organization in the first place. This is the detail that changes everything about how the earlier question should be understood. A gene responsible for something this fundamental cannot simply be switched off or deleted without consequence, because doing so would not just prevent a tail from forming later, it would interfere with the formation of mesoderm and the body axis itself, at a stage of development so early that almost nothing else in the embryo has been built yet to compensate for the loss. Researchers who have studied this gene closely in several different animals, including mice, cats, dogs, and fish, have found a consistent pattern that holds remarkably steady across species separated by tens of millions of years of separate evolution. When only one copy of the gene carries a disruptive mutation, the result is often exactly what the old name brachyury describes, a shortened or absent tail, with the rest of the body developing normally because the second, undamaged copy is still doing enough of the essential early work. This is why breeders of a well-known tailless cat and certain lines of dogs missing part or all of their tails can trace the trait back to a single altered copy of this same ancient gene, quietly carried and passed down through generations without harming the animals who inherit it. But when both copies of the gene are disrupted at once, leaving no working version left at all, the embryo typically does not survive far enough into development to be born. The same gene that shapes a tail, in other words, is also doing something the embryo cannot live without, and losing all of it is not a survivable option for evolution to select for. No matter how much a lineage might eventually stand to gain from a shorter or absent tail, part of what makes this gene so unforgiving to lose entirely is how far back its essential role stretches. Versions of it exist not only in mice, cats, dogs, and every ape, including humans, but in fish, whose own ancestors split away from the ancestors of every land vertebrate hundreds of millions of years before the tail loss discussed earlier ever happened. A gene doing the same basic job of organizing a body's early axis across animals separated by that much time and that much evolutionary distance is, by almost any standard biologists use, about as essential as a piece of genetic machinery can be, the kind of gene natural selection tends to protect rather than discard, because every lineage that has ever tried living without a working copy of it simply failed to continue. This changes the shape of the question left open a moment ago. A structure that seems, on the surface, expendable, a tail nobody in a tailless lineage appears to need anymore, sits downstream of a gene that is anything but expendable, one doing indispensable work at a stage of development that has nothing to do with tails at all. Whatever process eventually allowed an entire branch of the primate family to stop growing external tails could not have simply broken this gene the way a heavier, blunter mutation might. That kind of change, disabling the gene outright, would have carried too high a cost, threatening the exact same embryos it was supposedly benefiting, long before any of them ever reached the stage of life where having or not having a tail would matter to their survival at all. History offers no shortage of dead ends left behind by lineages that tried something close to this and failed, embryos that never made it far enough to be born, evolutionary experiments erased before they ever had a chance to matter. Evolution, in this specific case, was working with a gene it could not afford to lose, trying to remove one narrow function from it while leaving everything else about that gene fully intact. There is something worth sitting with in how precise a solution this actually required. Most of the genetic changes that shape visible differences between species involve altering how much of a protein gets made, or when, or where, in the body, small adjustments to timing and dosage rather than wholesale deletions of essential machinery. A change capable of quietly dialing down only the tail-building portion of TBXT's activity, late in development, at the specific site where a tail bud would otherwise form, while leaving every earlier, more fundamental use of the gene completely untouched, would have to be remarkably specific, almost surgical in how narrowly it acted. It could not be a change to the gene's core instructions, the part responsible for building the essential protein itself, because damaging that part would risk exactly the kind of early, whole-body failure already described. It would have to be a change to something more like the gene's scheduling, some adjustment capable of quietly withholding one particular output at one particular moment without touching anything the gene does earlier or elsewhere. Think of it less like removing a page from a book, and more like teaching a single reader to silently skip one paragraph on one specific page. while reading every other word on every other page exactly as written, leaving the book itself completely intact for anyone else who might need the whole thing. This is where the base of the spine, sitting quietly beneath the skin of whoever is listening to this right now, turns out to be carrying something far more delicate than a simple missing part. It is evidence of a gene that had to be edited with unusual precision, changed just enough to lose one specific late function, while every other part of what makes that gene essential to life remained exactly as it needed to be. Still active, still doing its older, more critical work in every single embryo, human ones included, at the exact same early stage. It has been doing that work for far longer than tails have mattered to this lineage at all. A blunt solution was not available here. Whatever actually happened inside this gene, in the ancestor shared by every tailless ape alive today, had to thread a very narrow needle, altering tail formation specifically without so much as touching the far more critical work the same stretch of DNA had been doing since the very earliest days after conception. What that precise change actually looked like at the level of the DNA itself is a different question from the one answered here, one that required scientists to look far more closely at the gene's exact structure than anything discussed up to this point, and what they eventually found there turned out to be stranger and more specific than a simple broken instruction. Picture a cell near the rear edge of a developing ape embryo as the TBXT gene is being read. Nothing there looks like a tail yet. There is no bone or visible extension, only a molecule being copied into a temporary message that will help decide what the rear of the body becomes. The gene itself has not been broken. Its main instructions remain capable of doing the early work described a moment ago, yet inside a space between those instructions sits a short stretch of DNA that was not always there. It belongs to the Alu family, often called jumping DNA because copies could appear at new genomic locations. This copy no longer jumps, it is fixed and inherited with the chromosome. But tens of millions of years ago, in an ancestor of every living ape, one such copy appears to have landed inside this crucial gene. Researchers call it AluY. It did not enter the protein coding sentence itself, but an intron, a stretch copied into the first RNA draft, and normally removed before the finished message is used. A direct strike into an essential coding region could have ruined TBXT. An intronic insertion could remain quiet enough for its carrier to survive and pass it onward. Comparing living genomes reveals the historical pattern. 1. The AluY insertion is found across hominoids, the group containing humans and apes, but not in the corresponding position in tailed monkeys. Its distribution places its arrival near the same deep fork described earlier, when the ancestral ape lineage separated from Old World monkeys roughly 25 million years ago. No fossil preserves the instant it occurred. The evidence lives in present cells, where the same sequence sits at the same address in a human, chimpanzee, gorilla, orangutan, and gibbon. Press 2 fingers against the blunt bone at the base of the spine, and the absence felt there can be traced to a small piece of inherited text still present beneath those fingers. AluY alone, however, was not enough to explain anything. It sits more than 500 DNA letters from the nearest coding block, too far away to act like a simple obstruction placed over an obvious cutting point. A search focused only on damaged protein instructions might have passed over it as background material. The clue appeared when researchers noticed another Alu sequence nearby, an older copy called AluSX1. This older element was already present in the broader monkey and ape lineage. It lies on the opposite side of exon 6 and faces in the reverse orientation. Imagine 2 short lines carrying closely related patterns, one written forward and the other positioned so that its copied message can face back toward the first. In the DNA, they remain separated, one in intron 5 and one in intron 6, with exon 6 lying between them. When the gene is transcribed, though, the cell produces a preliminary RNA message containing exons and introns together. The copied Alu sequences within that flexible RNA can bind to each other. As they pair, the RNA folds. A stem forms where they meet, while the material between them bends outward into a loop. Exon 6 is caught inside that loop. This is not the genome tying itself into a permanent knot, nor DNA being torn away. It is a temporary RNA shape formed while the cell edits the draft into a usable message. Normally, splicing machinery removes introns and joins coding exons in order. With the inverted Alu pair folded together, it may bring the closing edge of exon 5 close to the beginning of exon 7 instead. Exon 6 can then be passed over, leaving a shorter message in which exon 5 joins directly to exon 7. The word used for this is alternative splicing, because the same gene can be edited into more than one finished version. Human cells make both a full-length TBXT message and a version lacking exon 6. The ancient gene has therefore not been erased, and even exon 6 remains physically present in the DNA of every cell. It is simply omitted from some of the messages made from that DNA. The distinction is quiet but enormous. What the body inherited was not a deleted tail gene. It inherited a new way of reading a gene that still exists. At the base of the listener's spine, the coccyx marks the anatomical outcome of that altered reading, but the switch itself is not stored in the bone. It is reproduced whenever the relevant developmental cells copy TBXT and fold its early RNA into one shape rather than another. This proposal could not rest on resemblance and timing alone. AluY appears in the right animals and at roughly the right evolutionary moment, but genomes contain many changes that happen to travel with a lineage without causing its most visible traits. Researchers therefore tested whether the two Alu elements were actually responsible for the shorter TBXT message. In human embryonic stem cells encouraged to enter a developmental state resembling the early primitive streak, TBXT becomes active enough for its RNA products to be examined. Scientists used gene editing to remove AluY while leaving the surrounding gene intact. The version lacking exon 6 almost disappeared. They then removed the older AluSX1 instead, breaking the other half of the proposed pairing, and the exon-skipped version was again strongly suppressed. Neither result required deleting exon 6 itself. Removing either side of the inverted pair was enough to stop the editing pattern that hid it. In mouse embryonic stem cells, the researchers approached the question from the other direction. Mice do not naturally carry this same Alu arrangement in TBXT, so the team inserted the human AluY and AluSix1 elements on opposite sides of mouse exon 6. Once the cells were induced to express the gene, they produced the exon-skipped message. A separate design using a different pair of reversed, matching sequences also produced it, showing that the effect did not depend on some mysterious property unique to the name Alu. The important feature was the geometry, related sequences facing opposite ways, able to pair in the RNA and bend the exon between them out of the usual path. The molecular switch was no longer only something inferred from comparison. Its key movement had been reconstructed in living cells. The harder test was whether changing that message could change an actual tail. Here the evidence became more complicated and therefore more informative. Researchers made mice able to produce both the normal full-length form of TBXT and a form engineered to skip exon 6, broadly resembling the mixture of messages seen in hominoids. Some were born without an external tail, some with markedly shortened tails, and some retained long tails despite carrying the same engineered change. The variation was not a failure of the result. It pointed toward the importance of proportion. Tail development appeared to depend not merely on whether the shorter message existed, but on how much of it was present relative to the full-length message in the embryonic tail bud. Models producing more full-length TBXT tended toward longer tails. Models shifting the balance toward the exon-skipped form tended toward shorter tails or no external tail at all. In one engineered combination that raised the relative influence of the skipped form, every surviving mouse examined lacked an external tail. Yet mice carrying the inserted human Alu pair by itself produced only low levels of the shorter message in the relevant tail bud cells and showed no obvious adult tail loss. That limitation matters. It prevents the experiment from being reduced to the claim that one inserted Alu automatically removes a tail in every animal. The study instead supports a narrower and more careful conclusion. The Alu pairing can create the alternative message, and the balance between that message and the full-length form can powerfully alter tail development. Other genetic changes may have helped stabilize complete tail loss in ancestral apes after the AluY insertion first shifted that balance. Feel once more for the coccyx beneath the skin. What lies there is not the result of TBXT being switched fully off. The gene still performs its essential early work, and the body still begins the developmental path that briefly produces a tail. The alteration acts more like a reader trained to skip one passage while preserving the book, producing two related versions of the same instruction and changing their relative weight at a particular place and time. Exon 6 contributes to a regulatory region of the TBXT protein rather than its main DNA binding domain, so leaving it out may change how the protein influences other genes without destroying its ability to recognize DNA altogether. The detailed effects are still being studied, and the researchers themselves describe the AluY insertion as a plausible contributor rather than the only proven cause of ape tail loss. That caution does not make the mechanism less remarkable. It makes its scale easier to see. A short insertion in a non-coding interval did not need to invent a new organ, erase an ancient gene, or rewrite every cell. By folding a temporary RNA message differently, it may have adjusted how often one small coding block was heard, and that adjustment could reach outward from molecules too small to see to the final shape of the spine. The insertion survived one ancient individual, then a family, then a population, while climates shifted and ape lineages separated, until the same hidden arrangement arrived in every human body alive now. And AluY is not alone. The human genome is crowded with Alu copies and other repeated sequences, many lying inside introns, many facing neighboring sequences, in ways whose effects may depend on tissue, timing, and the fragile folding of RNA. Most will not silence a visible body structure. Some may do nothing detectable at all. But if one small pair inside TBXT was capable of helping turn a long external tail into the blunt coccyx resting beneath the listener tonight, how many other quiet switches are already folded into the body, active only in a certain cell, at a certain age, under conditions no one has yet thought to examine? Let one hand rest at the base of the spine again, not pressing hard now, only noticing how little information the skin gives back. Beneath the fingers, the coccyx feels like one blunt point, a single ending to a column of bones that seems as though it should be built the same way in everyone. If the skin were made transparent, however, and the soft tissue around it faded from view, that apparent simplicity would disappear. The coccyx is not always one solid piece, and it is not assembled from an identical number of elements in every human body. In most people, anatomists can identify 4 coccygeal vertebral segments. In others, they find 3. In still others, they find 5. All 3 counts fall within the recognized range of ordinary human anatomy. Someone resting in the next room may therefore carry one more small segment than the person listening now, while someone on the other side of the world may carry one fewer, and none of them would know it from the outward shape of the body. The difference is hidden beneath the same skin, at the same point below the sacrum, ending in nearly the same quiet curve. Detailed scans make the variation visible. On a side view, the first coccygeal segment appears broadest, sitting directly beneath the triangular sacrum. The elements below it become progressively smaller, until the lowest may look less like the familiar vertebrae of the back and more like a rounded bead of bone. In some adults, clear joint spaces remain between several of these elements. In others, some boundaries have fused so completely that the coccyx resembles the single bone most people imagine it to be. A study of adult CT scans found 4 segments in most of the people examined, but 3 and 5 were both present among otherwise normal coccyges. The researchers also found that fusion did not follow one universal pattern. The small structure beneath the listener's hand is therefore not a standardized relic stamped from a single rigid mold. It is a family of closely related endings, similar enough to share one name, variable enough that the exact arrangement cannot be known by touch alone. After the precise molecular mechanism just described, this may seem surprising. The Alu pairing within TBXT is shared across humans. The loss of an external tail is shared across humans. Every ordinary embryo follows the same broad movement from a visible early tail toward a body in which that extension has disappeared beneath the skin. Yet a shared developmental program does not require every finished bone boundary to land in exactly the same place. Bodies are not assembled by lowering a complete skeleton into waiting tissue. The vertebral column emerges through a time sequence in which early blocks of tissue form repeatedly along the head-to-tail direction of the embryo. Those blocks, called somites, contribute to the vertebrae and associated tissues. Each vertebra must then take shape through further growth, reorganization, cartilage formation, ossification, and, in the lowest region, extensive regression and remodeling. Near the middle of the back, the repeated pattern produces the large, recognizable bones that surround and protect the spinal cord. Near the farthest caudal edge, the same pattern is approaching a boundary. The early embryo has extended beyond the point where the adult spine will eventually stop, and the distal material must be reduced while the remaining segments are reshaped into the coccyx. A boundary created through growth followed by reduction does not need to resolve at precisely the same microscopic line in every person. Small differences in timing, segmentation, and remodeling can leave a slightly different count without restoring an external tail. The body with 5 coccygeal elements did not continue the ancestral construction very far. The body with 3 did not erase the past more completely. Both arrived at the same broad human form, but the last few marks left by the journey were not identical. Feel the tip beneath the fingers again and imagine the distinction hidden there. 3, 4, or 5. No sensation reveals the answer. There is no extra awareness in a 5th segment and no special emptiness where a 4th or 5th is absent. The variation becomes meaningful only when many bodies are placed beside one another through medical images or skeletal collections, and the assumption of perfect uniformity gives way to a population. Evolution has always worked at that scale. It does not produce a single ideal specimen and copy it without deviation. It acts through populations filled with bodies that resemble one another closely while differing in countless small measurements, angles, timings, and counts. Across generations, most of those differences remain quiet. They may not alter survival, movement, or reproduction enough for natural selection to favor one over another. They simply persist as a range, renewed whenever development turns inherited instructions into a particular body. The number of coccygeal segments appears to belong largely to this quiet kind of variation. 4 is most common, but common does not mean required, and a less common count is not automatically a return to an earlier evolutionary state. This distinction matters because it is tempting to arrange living people along an imaginary ladder. A person with 5 segments might be called more primitive, as if a longer ancestral tail were pushing through, while a person with 3 might be called more advanced, as if evolution had removed an additional piece. Neither description is justified. Evolution does not rank individuals by how closely one small bone resembles an ancestral condition. A person with 5 coccygeal segments is fully modern, born into the same tailless lineage and carrying the same broad developmental system as a person with 3. Nor does the present range show that humanity is steadily progressing toward fewer and fewer segments. To establish a change of that kind would require evidence across populations and generations, not a snapshot showing that adult anatomy varies now. What the range does show is quieter and more precise. The inherited program for ending the human spine permits more than one normal skeletal outcome. Tail loss became stable enough that no external tail is ordinarily completed, yet the structure left behind never became geometrically identical in everyone. There is another layer of variation that can easily be confused with segment number. Counting the elements and counting the spaces that still move between them are not the same task. A coccyx may have 4 developmental segments even when fusion has erased one or more visible joints. In another person, similar segments may remain partly separate. Fusion can continue as the skeleton matures, which means the appearance of the coccyx on a scan reflects both the pattern established early and the later history of bone joining to bone. An older-looking single piece is not necessarily built from fewer original elements than a chain with several visible gaps. The distinction resembles a row of stones whose edges can still be counted after they have been cemented together, except that living bone can blur some of those edges until only grooves or differences in shape reveal where one element met the next. This is one reason the coccyx has been easy to reduce, in ordinary speech, to a useless little bone. Its internal history is compressed into an object small enough to overlook. But look carefully, and it carries several layers of time at once. The embryonic tail that once extended farther, the developmental decision that reduced it, the individual count of the remaining segments, and the later fusion that may join them during growth. The point under the listener's hand is not one moment frozen in bone. It is the accumulated result of several moments, laid over one another until the boundaries are difficult to separate. A comparison with a tailed animal makes the scale clearer. In a monkey, the caudal vertebrae continue outward as a long series, each segment adding length and flexibility to an appendage that moves in open space. Differences in the number, length, and form of those vertebrae help create tails with different proportions and abilities. In a human, the external series has been reduced to a short internal remnant, so a difference of one small element does not create a visible tail or a new way of balancing through the world. It changes the hidden architecture of the ending, not the basic category of the body. The contrast is similar to two paths that stop at slightly different stones, while both remain far from the road that once continued beyond them. Humans with 3, 4, or 5 coccygeal segments are not divided into separate evolutionary directions. They occupy a narrow range inside the same ancient outcome. Still, the existence of that range softens the apparent sharpness of the boundary. Seen only from outside, the body seems to offer two clean possibilities: animals with tails and humans without them. Seen during development, and then inside adult anatomy, the border is less absolute. The embryo briefly extends a tail. The cells reduce it. The skeleton retains a coccyx. The coccyx retains a variable number of recognizable elements. The old program has been constrained, redirected, and shortened, but its final trace is allowed to settle in more than one arrangement. That is what it means to say the process was never closed into one perfectly fixed form. It does not mean tail loss is unfinished in the sense that ordinary humans are slowly moving toward a different species. It means development reaches a stable human result through a small field of permissible endings rather than through one exact count. Somewhere tonight, two people can lie in the same posture, feel the same blunt point beneath the skin, and carry different numbers of bones there without either body announcing the difference. The variation is silent, harmless in most cases, and almost impossible to discover without looking inside. Yet it leaves a narrow question resting at the farthest edge of the spine. If the stopping point can normally settle at 3 segments in one body and 5 in another, how close is the ordinary developmental boundary to the rare moment when the ancient extension does not stop beneath the skin at all? Imagine a quiet delivery room in the first minutes after a birth. The child is breathing, moving, and being examined in the ordinary way, but at the middle of the lower back, just above the crease between the buttocks. A small, skin-covered appendage extends outward, where the body usually ends in a hidden coccyx. It may be soft and narrow, it may hang without movement, or shift slightly when nearby muscles contract. The child may otherwise appear healthy, moving both legs with no obvious neurological problem, yet the structure is real, visible outside the body, occupying the place where every human embryo carried a tail weeks earlier. Cases like this appear in clinical reports written after medical examination. They are exceptionally rare, and the language used for them requires care. A visible appendage at the base of the spine is not automatically a persistent embryonic tail. Several very different conditions can create something that looks like one from the outside. A lipoma can form a projection of fatty tissue. A teratoma can grow in the sacrococcygeal region. An unusually elongated coccygeal vertebra can push outward. A meningocele or another form of spinal dysraphism can produce a skin-covered mass connected to deeper structures. Older clinical descriptions often gather these under the term pseudotail, because their resemblance to a tail comes from another developmental lesion, rather than the simple persistence of the distal embryonic appendage. The category traditionally called a true human tail is narrower. It usually describes a midline, skin-covered, boneless extension containing ordinary tissues such as fat, connective tissue, small bundles of skeletal muscle, blood vessels, and nerve fibers, without the vertebral chain that would make it a complete animal tail. In some reported newborns, imaging has found no underlying spinal dysraphism, and examination of the removed tissue has found no tumor and no continuation of the spinal column. That distinction answers the question left resting at the coccyx a moment ago, but it also changes it. The rare structure is not normally a row of extra vertebrae continuing the spine into open space. It is closer to the soft distal portion of an embryonic tail that failed to disappear completely. Place 2 fingers once more over the base of the listener's spine. In an ordinary body, the skin closes smoothly over the coccyx, leaving no outward sign of where the early extension ended. In these unusual newborns, a small amount of that distal tissue appears to have crossed the usual boundary and remained outside. One documented case involved a healthy newborn examined on the first day of life with a cutaneous appendage arising from the lumbosacral midline. Magnetic resonance imaging did not show spinal dysraphism. After the appendage was removed, microscopy identified skin over adipose and connective tissue, muscle bundles, blood vessels, and nerve bundles. There was no series of tail vertebrae hidden inside it. Under the terminology used by the clinicians, those findings supported the diagnosis of a true human tail. The name sounds more dramatic than the anatomy. It does not mean that an ancestral ape tail, with the bones, joints, and motor control of a monkey, returned intact, or that every tail-building instruction ran to completion. The more defensible interpretation is smaller. A late step in the normal regression of the embryonic tail may have remained incomplete, allowing a skin-covered remnant to persist after birth. During ordinary development, the early human tail contains a proximal portion associated with developing vertebral elements and a more distal portion made largely of soft embryonic tissue. As regression proceeds, the outer extension disappears, and the remaining proximal skeletal elements are remodeled into the coccyx. If the distal soft tissue does not fully regress, the result can resemble the narrow appendages placed in the true tail category, even though the deeper vertebral sequence has still been reduced in the usual human way. This is why calling the event evolution in reverse would be misleading. Evolution changes inherited traits across populations and generations. A congenital variation in one child is a developmental outcome. It may reveal what an old developmental pathway is capable of producing when its timing changes, but it does not move that child backward through ancestry. The child's entire genome, skeleton, brain, limbs, and physiology remain those of a modern human. The appendage does not place the child between humans and tailed monkeys, and it does not represent a separate stage on a ladder that biology does not contain. It shows only that a process usually completed before birth can on very rare occasions stop at a different point. The coccyx beneath the listener tonight and the soft appendage described in a clinical report are therefore not two opposite designs. They are two outcomes near the final edge of the same developmental sequence, one overwhelmingly common and internal, the other exceptionally uncommon and visible. Even the words true and false can make this seem cleaner than it is. Clinical classifications have changed as more cases and better imaging have become available. A surface appendage may look harmless, while an MRI reveals a tethered spinal cord, a split in the vertebral arch, a fatty connection, or another hidden abnormality. Conversely, a structure may appear striking from outside while detailed imaging finds no connection with the spinal canal and no malformation beneath it. Histology adds another layer by showing which tissues actually occupy the appendage. For that reason, physicians cannot safely classify a newborn's caudal appendage by shape alone. A careful neurological examination and imaging of the spine are important even when the child moves normally, because some forms of occult spinal dysraphism are not immediately obvious at birth. This medical caution should not be confused with alarm. It means only that similar outward shapes can arise through different internal routes, and those routes matter for treatment. The visible form is the beginning of the investigation, not its conclusion. For the same reason, reports in older literature must be read with some restraint. Before modern imaging, a physician could describe the length, softness, movement, and skin covering of an appendage, but could not always see whether a fine tract connected it to the spinal canal. Some cases, historically called true tails, might be classified differently today. Because it is rare, knowledge comes mainly from case reports rather than large studies. The evidence supports the existence of benign boneless caudal appendages without underlying spinal dysraphism, but it does not support treating every photograph projection as the same biological event. The difference between a persistent embryonic remnant and a tail-like sign of another anomaly has to be demonstrated, not assumed. Return again to the newborn in the quiet room, now without the labels. What exists is a small piece of living tissue where ordinary human development usually leaves smooth skin over 3, 4, or 5 coccygeal segments. Its cells are not ancient cells. They were made during that pregnancy, using the same chromosomes and chemical signals as the rest of the child's body. What is ancient is the developmental root that gave those cells a place to form. The root begins in every human embryo, then is normally narrowed, dismantled, and hidden. In this rare body, the final disappearance may not have been complete. That is enough to make the boundary between presence and absence feel less absolute, but not enough to say the whole ancestral organ has returned. Developmental systems often work this way. They are sequences of signals, thresholds, growth periods, and stopping times. A small shift does not need to rebuild everything. It may preserve only the last soft trace of a structure, while every deeper transformation proceeds normally. The difference between no visible appendage and a short one can therefore be smaller at the molecular level than the outward contrast suggests. A slight change in when cells stop dividing, when programmed cell death occurs, or how tissues are remodeled may be sufficient, although the precise cause in any individual case is often unknown. It would be tempting to connect every such birth directly to the Alu arrangement in TBXT, but the evidence does not permit that. The Alu pairing is shared by essentially all humans, while these appendages are extraordinarily uncommon. If that common arrangement were sufficient by itself, the condition would not be rare. Other developmental events, genetic variants, or local differences in signaling must be involved, and in many reported children they have not been identified. The case, therefore, does not overturn the mechanism already described. It shows that the final shape of the caudal region depends on more than one control point. TBXT and its splice forms help set the broader conditions for tail reduction, while apoptosis, tissue remodeling, neural tube closure, vertebral development, and other pathways determine how cleanly the outer structure disappears. The blunt point under the listener's fingers is the usual result of all those systems converging. A persistent soft appendage is evidence that their convergence can occasionally differ by a very small amount. Across a whole human population, such an event is too rare and too inconsistent to create a new anatomical norm. It appears in one family, is examined, and usually ends as a medical record rather than spreading through generations as a stable trait. That separates it from the ancient event in the ape ancestor, where a heritable change survived reproduction and became shared across an expanding lineage. One is a rare variation in development, the other is evolutionary change carried through population time. Their outward relationship to a tail may look similar, but their histories are not the same, and that difference opens a wider comparison. Humans are not the only mammals in which the length or presence of a tail can change. In some cats and dogs, taillessness has become a repeatable inherited trait rather than a singular developmental exception. Those animals reach a similar outward result through alterations connected to the same ancient gene family discussed earlier, but not through the delicate Alu pairing that appeared in the ape lineage. If one gene can be touched in different ways, in unrelated mammals, and repeatedly alter the lowest edge of the spine, what is it about TBXT that makes the tail such a reachable target while the rest of the body must remain intact? Picture a Manx cat walking across a quiet room, no long tail following its body. One cat may have only a hollow where a tail begins, another a small bony bump, and another a short stump. Some born into the same breed carry a tail approaching ordinary length. These differences are present from birth, not caused by injury or removal, and unlike the rare, soft human appendage described earlier. In the Manx cat, the vertebral series itself is shortened, sometimes until no external tail remains. When researchers traced this trait through cat families and then read the relevant DNA, they arrived at the same developmental gene already sitting beneath the human story, TBXT, historically called T in animal studies, and the source of the brachyury protein. The resemblance ends there, because the molecular root in these cats is not the Alu mechanism found in apes. Nothing folds around exon 6 to make the cell occasionally skip it. The Manx mutations lie inside the protein coding instructions. 3 of the variants identified in one major study were deletions of a single DNA letter. A 4th was a small event in which a short stretch was duplicated while nearby material was deleted. Each disturbed the reading frame, the 3-letter rhythm by which a cell translates DNA information into amino acids. Once that rhythm shifts, every instruction downstream can be misread until a premature stop signal appears. The predicted result is a shortened brachyury protein missing its normal carboxy terminal region, a much more direct disturbance than the alternative splice pattern inherited by humans and other apes. Yet the cat usually carries only one altered copy. The other copy remains intact, supplying enough working brachyury for the embryo to form most of the body successfully. Tail development is less forgiving of the reduced dosage. Geneticists call this haploinsufficiency. One functioning copy is sufficient for much of development, but not sufficient to produce the full result normally supported by 2. The word sounds abstract until the cat crosses the room again. The shortened or absent tail is the visible measurement of a protein supply that fell below what the caudal region required. In a study with complete genetic and physical information from 130 Manx cats, 78 of 82 cats with shortened tails carried one of the mutant T alleles, while 47 of 48 full-tailed cats carried only the usual forms. Different family lines carried different mutations, suggesting several independent disruptions rather than one variant spreading into every tailless cat. Even cats carrying the same altered allele did not all finish with the same tail length. Within pedigrees, kittens could range from no visible tail to a short stump or a much longer tail. Other genes, local developmental conditions, and small differences in expression appear to modify how strongly the mutation is expressed. The gene sets the caudal region close to a threshold, but it does not dictate a single length with mechanical certainty. Rest a hand again over the listener's coccyx and compare the roots. In a human cell, both the full-length TBXT message and the exon-skipped form can be produced from an intact gene. In a Manx cat, one chromosome may carry a coding frameshift predicted to truncate the protein, while the other carries a working copy. Both arrangements leave enough essential activity for many embryos to survive, yet both alter what happens at the lowest edge of the spine. They approach the same anatomical neighborhood from different molecular directions. Researchers found no Manx cats carrying 2 disruptive copies or 2 different damaging variants. That absence supports the expectation that losing too much brachyury activity is not compatible with normal embryonic survival. Among surviving Manx cats, The most extensive caudal reduction can also be associated with abnormalities of the sacrum, spinal cord, hind limbs, bladder, or bowel, though many short-tailed cats do not have these problems. The same narrow bargain seen in ape evolution appears again. Enough change to shorten the posterior extension, but not so much that the early body plan fails altogether. Now move to a litter of dogs. A Pembroke Welsh Corgi, Swedish Vallhund, or member of several other breeds may be born with a natural bobtail of variable length. Here again, one altered TBXT copy can change the tail. But the common canine mutation is different from both the Manx frameshifts and the ape Alu Insertion. It is a substitution of a single DNA letter, known as C189C to G, which changes one amino acid in the brachyury protein, replacing isoleucine with methionine at position 63. That location falls within the T-box region, used by the protein to recognize and bind DNA. The canine protein is not simply shortened at its far end, and its RNA does not skip the same exon as ape TBXT. One component inside the protein's conserved binding region has been changed. The result is again dosage sensitive and variable. Dogs with one copy tend to have congenital bobtails of differing lengths, while long-tailed relatives lack the variant. Researchers tested 360 dogs from 23 breeds in which natural short tails occurred. In 17 breeds, where the C189C to G substitution was present, it matched the short-tail phenotype. In 6 other short-tailed breeds, however, the known substitution was absent, and the investigators did not find another coding mutation in T that explained the trait. That negative result is as important as the positive one. TBXT is a repeated route to a short tail, not the only route. Development contains many genes capable of influencing vertebral number, tissue growth, and caudal extension, and different breeds can reach a similar outward appearance through different inherited changes. The dog study also found no animals carrying 2 copies of the common substitution. When 2 naturally short-tailed Swedish Vallhunds were paired, their litters were smaller on average than litters from long-tailed parents, supporting the interpretation that embryos inheriting 2 copies may often fail to survive. The evidence is indirect rather than an observation of every missing embryo, so the conclusion is properly described as strongly suggested, not proven, in each pregnancy. Still, it fits the same pattern found in mice, cats, and the earlier work on TBXT. The rear of the body responds visibly when activity is reduced, while a larger reduction can cross a boundary the whole embryo cannot tolerate. Beneath the listener's fingers, the human coccyx sits at the human version of that boundary, but it was reached through an older and more indirect change. Place the 3 lineages beside one another without pretending their histories were the same. In the ancestral ape, an AluY insertion paired with an older AluSX1 element altered RNA folding and encouraged exon 6 to be omitted from some TBXT messages. In Manx cats, several independently arising coding mutations shifted the reading frame and prematurely shortened brachyury. In many naturally bobtailed dog breeds, a single amino acid substitution altered the conserved DNA binding region of the protein. These changes did not descend from one tailless mammal that passed the trait to all 3 groups. The ape insertion occurred in a primate ancestor tens of millions of years before modern humans. The feline mutations arose much later in domestic cat lineages. The canine substitution followed its own history and was then carried through multiple breeds. Similar anatomy appeared more than once because separate populations altered the same developmental control point in different ways. This is convergent change at the level of a gene, but even that phrase requires restraint. Domestic cats and dogs have lived under human breeding choices, so the spread of short-tail variants in those populations cannot be described as natural selection alone. A mutation may have appeared without intention, but people could preserve and amplify the appearance through selective breeding. In the ancient ape lineage, no breeder chose the outcome, and the pressures that allowed tail loss to spread remain uncertain. The forces moving the variants through populations were different. What converged was the genetic vulnerability they encountered. Why should TBXT be encountered so often? One reason may be its position at a developmental threshold. Because brachyury helps organize the posterior body and tail bud, a modest decrease or altered form can produce an immediate change at the farthest vertebrae. The tail is a repeated structure added in the caudal direction, shortening the period of extension or reducing the supply of a key regulator can remove several terminal segments without redesigning every tissue in the body. TBXT is therefore capable of translating a small molecular change into a large visible difference. At the same time, its essential early roles place a hard limit around that flexibility, break both copies too severely, and the embryo may not survive. Reduce function in the wrong cells or at the wrong time, and the result may include spinal or sacral abnormalities rather than an otherwise healthy short tail. Evolution and breeding can work only inside the narrow interval between no visible effect and too much damage. This helps explain why different lineages repeatedly touch TBXT, but it does not mean the gene is uniquely destined to control every tail. More than 100 genes can produce shortened or absent tails when altered in laboratory mice, and the short-tailed dog breeds lacking the common T mutation demonstrate that other roots exist in living animals. TBXT stands out because the tail is unusually sensitive to its dosage and structure, not because nature has only one switch available. Feel the blunt human coccyx once more. The comparison with cats and dogs makes it look less like an isolated relic and more like one stable point within a broad landscape of possible caudal outcomes. A looped RNA message, a frame shift, or one changed amino acid can move a body to a different place in that landscape. But none of these changes erases the lowest spine completely. Some remnant remains because the rear of the body cannot simply be cut away from the developmental systems that organize it. And if the coccyx has been retained across millions of years after the external tail vanished, perhaps the reason is not only that evolution could go no farther without risk. Perhaps the small bone beneath the listener is no longer merely remaining after an abandoned program. What, quietly, is it still doing there? Without changing position, notice where the chair is holding the body now. Beneath the softness are two rounded projections of the pelvis, called the ischial tuberosities, the bones people mean when they speak of sitting bones, and in an upright posture they carry much of the load. The coccyx sits farther behind them, tucked into the lowest fold of the body, small enough that it can seem removed from the work of sitting altogether. Now imagine, without needing to move, letting the pelvis roll backward and the chest settle toward the back of the chair. The contact map changes. Pressure that had been concentrated beneath the sitting bones shifts rearward through the surrounding tissue, toward the sacrum and the coccyx. Measurements made with pressure-sensitive seats have recorded this redistribution under certain combinations of tilt and recline, and anyone who has ever landed hard on the tailbone knows, without an instrument, that the point is capable of meeting force. It is not a third leg, holding the body up in the same way the two sitting bones do, and the exact share of the load varies with anatomy, cushioning, and posture, but it is not floating untouched inside the pelvis either. At this very moment, while the listener rests, an ancient remnant at the spine's lowest point is participating in the quiet mechanics between a living body and the surface beneath it. Even the bone itself is less fixed than its blunt shape suggests. In many people, small joints remain between some of its segments, or between the coccyx and the sacrum above it. When a person moves from one posture to another, the coccyx may flex by a modest amount, changing its angle under load. Dynamic radiographs, made by comparing images taken in different positions, reveal movements that an ordinary picture of a skeleton cannot show. Inside a living person, it belongs to a system that bends, tightens, cushions, and responds. Excessive movement can become painful in some people, while other coccyges move very little, so there is no single mechanical pattern shared with mathematical precision by everyone. The important point is quieter. The remnant does not merely occupy space. Force reaches it, tissues pull on it, and its position changes with what the rest of the pelvis is doing. To see the rest of its work, Let the imagined view pass inward through the seat, skin, and layers of muscle until the pelvis appears not as a solid bowl of bone, but as a ring with a flexible opening at its base. Across that opening lies the pelvic floor, a broad arrangement of muscle and connective tissue often compared with a hammock. The comparison is useful as long as the hammock is not pictured as slack cloth. These muscles hold a changing, active tension. They help support the organs above them, contribute to the control of the openings that pass through them, and adjust as pressure rises within the abdomen. Several of those fibers find an attachment at or near the coccyx. Portions of the levator ani reach toward it through the tissue behind the anal canal. The coccygeus muscle runs toward the sacrum and coccyx from the sides of the pelvis. A fibrous seam called the anacoccygeal body links the region of the anal sphincter to the tailbone behind it. Around them, ligaments connect the coccyx to the sacrum and to other landmarks of the pelvis, while fibers of the gluteus maximus, the large muscle shaping each buttock, also attach along the back and side of the lower sacrum and coccyx. The small bone beneath the listener is therefore not sitting alone like a pebble left in an empty pocket. It is a knot within a web of tension. When posture changes, the pull changes. When the pelvis receives pressure from above or contact from below, this network helps manage the result. None of that requires the coccyx to behave like the long tail it once preceded. There is no row of powerful caudal muscles moving it freely through the air, no counterweight adjusting the body on a branch. Its old function has been reduced beyond recognition, but the material left behind has been incorporated into another arrangement. This is why the word vestigial can be misleading when it is heard as a polite scientific synonym for useless. In biology, a vestige is evidence of a structure whose ancestral form and main role have been greatly reduced or altered. It does not have to be idle. An old structure can lose the task that originally shaped it while retaining smaller roles, or becoming useful in a new mechanical setting. The human coccyx does not balance a body running across branches, yet it offers anchorage to tissues that help stabilize the bottom of a body sitting in a chair. The distinction matters, because evolution rarely clears a workbench and begins again with clean materials. It changes what is already present. A structure inherited from one way of life may be shortened, folded inward, and recruited into another. Its history still visible in its shape even while present-day muscles treat it as an ordinary place to attach. Feel again, only in imagination, the small blunt point beneath the skin. More than 20 million years separate it from the population in which the external ape tail disappeared. Across that time, the coccyx did not remain mechanically frozen. In the human lineage, habitual walking on two legs altered how the trunk balanced over the hips, and the pelvic floor had to support abdominal and pelvic contents above an opening, oriented beneath an upright body. Childbirth placed its own unusual demands on the same passage. It would be too simple to claim that the coccyx was preserved for any one of these later human functions, because the bone was already present in tailless apes long before human posture or human childbirth existed. It would also be too simple to imagine that every detail around it is an untouched remnant of the forest. The safest picture is one of continued integration, an inherited endpoint of the spine becoming entangled, generation after generation, with the mechanical needs of each descendant body. During defecation and childbirth, the pelvic outlet must accommodate changes in shape and pressure, and the small joints around the coccyx may allow some yielding. During ordinary sitting, the same region can receive contact and load. During contraction of the pelvic floor, attached tissues use it as one of several anchors. Not the only anchor, and not an irreplaceable command center, but a real participant. This makes the surviving bone more interesting than either of the labels commonly given to it. Calling it a hidden tail says where it came from, but not what it does now. Calling it a useless leftover acknowledges its reduced form but misses the living network attached to it. The coccyx is both a remnant and a working piece, an ancestral base repurposed without ever being detached from its history. The gene mechanisms described earlier help explain how the external extension could be suppressed without destroying the developmental systems that build the rear of the embryo. The anatomy beneath the listener adds the other half of the paradox. Evolution did not need to erase every final trace once the tail was gone. What remained could continue as the lowest point of the spine, available for muscles and ligaments to pull against, while the rest of the body reorganized around it. This should not be mistaken for foresight. No ancient mutation anticipated chairs, upright humans, or a future pelvic floor and saved the coccyx in preparation. Each generation simply inherited a body assembled from the previous one, and any workable new arrangement had to be made from those existing parts. What survives can acquire consequences its earliest form never had. What looks unfinished from the perspective of an engineer can be thoroughly woven into the organism from the perspective of development. Somewhere in a living tailed monkey, muscles at the base of the tail transfer force between pelvis and a chain of vertebrae extending outward. In an ape, that chain ends almost as soon as it begins, but the base is still surrounded by muscle, ligament, nerves, blood vessels, and connective tissue. The appendage vanished. The junction did not. In humans, that junction now lies precisely where the weight of the upper body, the tension of the pelvic floor, and the pressure of a seat can meet. Return once more to the body, hearing these words. Perhaps it has shifted since the opening sentence without noticing. Perhaps the pelvis has rolled a little farther back as sleep approaches, bringing the tailbone region more firmly toward the mattress or chair. Under the listener, a remnant older than every member of the genus Homo is doing something entirely present, receiving pressure, giving fibers a place to attach, moving slightly within a network that never stops adjusting. It is not performing the spectacular work of a prehensile tail. Its work is so quiet that most people notice it only when the region hurts, and otherwise pass decades without thinking of it once. Yet silence is not absence. The oldest visible consequence of tail loss now helps a tailless body support its contents, close its pelvic openings, and position itself today. The gentle irony is that the feature called a leftover has outlived the external structure it belonged to by more than 20 million years, and throughout that immense interval, it has never been outside the body's current business. But this creates a different historical problem. The listener may naturally call it the human tailbone, and imagine its transformation as an episode in human evolution. The dates do not allow that. By the time anything that could be placed on the human branch separated from the ancestors of chimpanzees, the external tail had already been absent for tens of millions of years. No early human watched it shorten. No population belonging only to humanity crossed from tail to tailless. The working coccyx beneath the listener was inherited with that ancient change already complete. If losing the tail was never a specifically human event, then which portion of the story, if any, truly belongs to humans alone? Imagine kneeling beside a shallow excavation in eastern Africa while the last heat of the day rises from the sediment. A brush moves slowly across a patch of pale bone no wider than a fingernail. Nothing about the fragment announces a species, a posture, or an age. This is what the human fossil record usually looks like at close range. Not an orderly procession of complete skeletons, but teeth without jaws, jaws without skulls, portions of pelvis compressed by stone, and isolated limb bones separated from the individuals who once used them. A coccyx is especially easy for time to remove. Its vertebrae are small, its joints can separate, and the scavenging, weathering, burial, and geological pressure that precede discovery do not preserve every bone equally. If the search for the moment humans lost their tails depended on finding a perfect sequence of fossil tailbones, the answer might remain unreachable. But the deeper problem is that the search would be taking place on the wrong branch of the family tree. Move backward from the person listening now, past the first Homo sapiens, past earlier members of Homo, past Australopiths walking through African landscapes more than 3 million years ago, and past Ardipithecus with its mixture of climbing and upright movement around 4.4 million years ago. Continue toward the uncertain cluster of fossils near 6 or 7 million years old. close to the time the ancestors of humans separated from the ancestors of chimpanzees and bonobos. Even there, at the earliest boundary researchers can reasonably discuss as belonging to the human lineage, the external tail is already ancient history. The hominoid lineage had become tailless roughly 25 million years ago, many millions of years before that later division. Place 2 marks on an imagined timeline beneath the listener. The older mark records the divergence of apes from the tailed Old World monkey line and the inferred loss of the external tail. The younger mark records the much later separation of the human and chimpanzee lineages. The coccyx under the body crosses the younger mark without changing from present to absent. It arrives already shortened, already enclosed, already inherited. No population on the human side begins that journey with tails and finishes it without them. The first individuals anyone might call hominins did not stand at the edge of a new anatomical subtraction. They began with the subtraction behind them. This distinction is easy to miss because evolutionary stories are often told backward from the body, asking the question. A human looks at a monkey, notices the tail, and naturally asks when humans lost theirs. The grammar assigns the event to humans before the evidence has even been considered. A more accurate question would ask when the ancestors shared by humans and all other apes lost the external tail, because the inheritance belongs to that much older population. The same absence passed through the ancestors of gibbons, orangutans, gorillas, chimpanzees, bonobos, and humans, branching repeatedly while remaining recognizable in every descendant line. Bring 2 infants into the imagined comparison, one human and one chimpanzee. Neither begins with an external tail. Move back 1 generation, then another, and the shared condition does not converge within human history. It continues through a common ape inheritance far older than either species. The small blunt bone beneath the listener therefore marks kinship before it marks humanity. Fossils from the human branch can reveal what later evolution did with a body that was already tailless. A femur can carry evidence of how weight passed through a hip. A pelvis can preserve clues about upright walking and birth. Foot bones can show a changing relationship between grasping and propulsion. A skull can place the opening for the spinal cord beneath the head in a way connected with habitual posture. These are transformations that unfolded within hominin history, unevenly and in combinations that researchers still debate. Tail loss is different. It does not appear among them as one more transition waiting to be arranged between one early human species and the next. By the time Australopithecus afarensis crossed volcanic landscapes between roughly 4 and 3 million years ago, the missing tail was not an innovation of its population. When Homo sapiens emerged only a few 100,000 years ago, the Alu insertion implicated in the ape pattern had already been copied through an almost unimaginable depth of generations. Species names can make those generations look like rooms separated by walls. One room for Ardipithecus, another for Australopithecus, another for Homo. Real populations did not experience themselves that way. Offspring resembled parents, populations divided and mixed, and small differences accumulated without anyone crossing a labeled threshold. An inherited feature could flow through every one of those changes without being reinvented. The absence of a tail did exactly that. It moved through the human lineage, not as a fresh adaptation each time the environment changed, but as background anatomy, something already settled before the traits that later seemed distinctly human began to assemble. Return to the chair or bed for a moment and notice how strange that makes the coccyx feel. It lies inside a body shaped by upright walking, a body with its own proportions of spine, pelvis, and legs. Yet the reason there is no long chain of caudal vertebrae behind it is not tied specifically to walking across a savanna carrying tools, controlling fire, speaking, or building cities. All of those appeared far too late to cause the original loss. Even habitual bipedalism in hominins cannot be treated as the event that removed the tail, because the tail had vanished before the hominin form of bipedalism began. The missing appendage may have influenced what kinds of movement became possible later, and a tailless ape body may have provided a different starting condition for upright posture, but sequence is not the same as cause. A trait that came first cannot be explained by an adaptation that appeared millions of years afterward. This is also why a gap in the human fossil record cannot hide a secret human tail loss episode. Suppose tomorrow an unknown hominin were found from a poorly sampled interval 5 million years ago. Its skeleton might transform ideas about locomotion, diet, or relationships among early populations. Finding an external tail on it, however, would conflict not only with what is known from other hominins, but with the shared anatomy and shared genetic signature of the entire ape family. The evidence would demand extraordinary checking because it would imply that a lineage regained a long-lost structure or that its place on the family tree had been misunderstood. The absence of such a fossil is not the main argument. The main argument is the pattern already present across living species and their genomes. A specific AluY insertion near an older Alu element in TBXT is shared by hominoids and absent from the tailed monkeys used for comparison. That distribution points toward a change in a common hominoid ancestor, not towards separate changes in humans, chimpanzees, gorillas, and gibbons. Fossils from early apes add a compatible anatomical record, with tailless forms present long before recognizable hominins. The independent clocks agree, even though neither is complete. Comparative anatomy places the absence deep within apes. Genetics identifies a shared molecular event on that deep branch, and fossils place tailless ape bodies millions of years before the human line began. What remains uncertain is the precise ecological pressure that allowed or favored the change, not whether Homo sapiens lost a tail of its own. There is a quiet correction hidden here. The phrase human tailbone is anatomically convenient, but historically possessive. It can make the Coccyx sound like a private relic from a uniquely human transformation. In reality, a chimpanzee, gorilla, orangutan, or gibbon carries its own version of the same ancestral reduction. Human evolution received that reduction and then modified the surrounding body. The pelvis became reorganized for sustained bipedal walking. The lower spine took on human patterns of curvature and loading. The pelvic floor worked within the unusual pressures of upright posture and human childbirth. Those later developments may have changed how the Coccyx participates in daily mechanics, just as the previous journey suggested, but they did not create the Coccyx by removing an external human tail. They remodeled a house whose rear wall had been shortened long before the occupants arrived. Feel the pressure beneath the listener again. The bone there belongs simultaneously to two histories, an ape history that explains why the external tail is absent, and a later hominin history that helps explain the particular body now resting upon it. Confusing, those histories compresses more than 20 million years into one misleading image, as if a nearly human ancestor once stepped upright and its tail then faded away. No known fossil records such a scene, and the branching order leaves no place where it is required. The ancestors who first walked habitually on two legs were already tailless apes. The ancestors who began shaping stone inherited a Coccyx. Neanderthals inherited it. Every population of Homo sapiens inherited it. Across migrations, ice ages, bottlenecks, and encounters between human groups, the external tail was not being lost again and again. The old genetic arrangement was simply being copied, quietly, as all the more recent human differences accumulated around it. So what truly belongs to humans in this story? Perhaps not the anatomical disappearance at all. Perhaps the distinctly human event occurs much later, when one tailless ape learns to extract DNA from cells, align its sequence with those of other primates, and recognize inside a living genome the trace of a change that happened before its own lineage existed. A fossil Coccyx can reveal shape when it survives. A sequence can preserve ancestry even when no bone from the original population has ever been found. The listener carries that sequence now, not sealed in a museum drawer but repeated inside living cells throughout the body. If the decisive event lies too far back to appear in the human fossil record, how can a small sample taken from a person alive today reach farther into the past than any shovel. Set the shovel aside and imagine a cotton swab moving once along the inside of the listener's cheek. The pressure would be barely noticeable, yet the damp tip would lift away thousands of cells, each one too small to see without magnification. Inside their nuclei would be living copies of a genome carried through every generation described so far. In a laboratory, chemicals can open those cells and separate their DNA from proteins, membranes, and the other crowded material of life. The molecules are far too long to be read all at once by many sequencing methods, so they may be broken into smaller fragments and prepared in enormous numbers. A machine does not look at the fragments and see printed letters. Depending on the technology, it detects physical or chemical signals associated with the bases. Then software translates those signals into the familiar symbols A, C, G, and T. The resulting stretches are called reads. Some are short, some can now extend for thousands or far more bases, and overlapping reads can be assembled or aligned against a reference until a much longer sequence emerges. It is a little like receiving millions of torn strips from many copies of the same immense manuscript and using their overlaps to work out where each strip belongs. Repeated DNA can make locations look nearly identical, so researchers compare overlapping copies and assign confidence to what instruments report. One nucleated human cell contains the genome in principle, but practical whole genome work commonly uses DNA from many cells or amplification to provide enough material and repeated coverage. What matters is that the historical trace exists in every ordinary cell, carrying a nucleus, including cells collected from the cheek of the person listening now. Once the sequence has been reconstructed, it does not arrive with labels saying tail, ape, or 25 million years. It is simply an order of roughly 3 billion base pairs, most of which would reveal little about tail loss if examined alone. The past becomes visible only when one genome is placed beside another. Imagine a computer screen holding the same neighborhood of TBXT from a human, a chimpanzee, a gorilla, an orangutan, a gibbon, and several tailed monkeys. Algorithms line up stretches that descend from the same ancestral region, allowing for bases that have changed, sequences that have been deleted, and insertions that make one row longer than another. Across much of the display, the letters resemble one another because all of these animals inherited them from older primate ancestors. Then one row reaches the location discussed earlier, and the comparison becomes uneven. The AluY sequence appears in the hominoids at the corresponding location, while the tailed monkeys lack it. Nearby, the older Alu element is present more broadly. No researcher had to recover a 25-million-year-old cell to see this pattern. The descendants carried it forward. If humans alone contained the insertion, it could have arisen after the human lineage separated. If humans and chimpanzees shared it but orangutans and gibbons did not, it would point toward another interval. But its presence across living hominoids places its origin deeper, before those descendant lines separated from one another, and after their lineage diverged from the tailed monkeys that do not carry it. The family tree turns distribution into time. Not an exact calendar date printed inside the sequence, but a bounded historical inference strengthened by genetic estimates of divergence and by fossils that help calibrate the branches. This is how a living cheek cell can reach farther back than the shovel from the previous scene. The arrangement copied inside it is ancient. Its DNA was not preserved unchanged as one molecule across 25 million years. Each generation made new molecules from older templates, and mutations accumulated along the way. Yet the insertion remained recognizable in the same genomic neighborhood, passed from parent to offspring while populations divided into species. The material is new. The relationship among the sequences is old. Feel the coccyx against the chair or mattress again. Bone provides the outward clue that something has been reduced. Comparative sequencing supplies a second record inside the same body, one that can remain informative even when the original bones were scattered, dissolved, or never fossilized. In this sense, the genome behaves like a fossil that was never buried. But the comparison needs care. A stone fossil is a physical remnant of one organism at one moment. A genome is a continually copied descendant of earlier genomes, shuffled by recombination and revised by mutation in every lineage. It is closer to a manuscript recopied by generations of scribes than to an object sealed motionless in rock. Lines can be lost, new passages can be inserted, repeated phrases can confuse reconstruction. Natural selection can preserve some changes while removing others, and random chance can allow a sequence to spread or vanish without leaving a clear explanation. Much of genomic history has been overwritten beyond confident recovery. Even when an old difference is found, sequence comparison alone does not automatically reveal what it did. The AluY insertion became persuasive not merely because it occupied an interesting position, but because researchers connected it to altered processing of the TBXT message and then recreated related outcomes in engineered mice. The comparative pattern located a candidate historical change. Experiments tested whether that change could affect tail development. Neither approach carried the full answer alone. This distinction protects the story from becoming too neat. Modern sequencing can expose a trace that no archaeologist could excavate, but it does not turn every stretch of DNA into a sentence with one obvious meaning. A colored band on a genome browser may represent years of work deciding whether reads were aligned correctly, whether a sequence is genuinely shared, whether its activity differs among tissues, and whether an observed association is causal or merely nearby. Repetitive elements such as Alu sequences are especially demanding because the genome contains so many related copies. Short reads may fit more than one location, while longer reads can sometimes anchor a repeated sequence to unique DNA on either side and reveal its context more clearly. Better instruments expand what can be seen, but interpretation still depends on comparison, developmental biology, statistics, and experiments in living systems. Return once more to the body from which the cheek cells came. The sequencer does not need to know that this body has a coccyx. It reports bases. The computer does not need to understand what a tail feels like in motion. It aligns sequences. Meaning appears when those neutral outputs are connected back to anatomy, embryonic development, and the branching history of primates. The small bone under the listener, the temporary tail in a human embryo, the Alu arrangement inside TBXT, and the tailless bodies of all living apes become independent observations that point toward the same old change. The strength comes from convergence, not from one instrument claiming to show the past directly. Only recently in human history has such convergence been possible at this scale. For almost all the time Homo sapiens has existed, nobody could read a single base of DNA. The first broadly useful sequencing methods arrived within living memory, and the first human reference sequence required an international effort measured in years. Once human DNA could be compared with complete or increasingly detailed genomes from chimpanzees, gorillas, orangutans, gibbons, monkeys, mice, and many other animals, features that once looked like meaningless repetition acquired positions on a family tree. Databases and genome browsers made it possible to move across chromosomes without touching a bone, to ask which sequence appears in which species, and to see whether a supposedly human feature is actually shared across a much older lineage. More recent sequencing methods can span longer and more repetitive regions, while comparisons across hundreds of primate genomes provide a finer background against which an unusual insertion can be recognized. The archive was present in bodies long before the tools capable of reading it existed. The difference today is not that the genome suddenly began recording history. It is that one species finally built instruments and mathematical methods sensitive enough to notice the record. This may be the most specifically human turn in the entire journey. Humans did not lose the tail as humans, but humans became able to discover how an ancestral ape lineage may have lost it. A change copied silently for more than 20 million years eventually encountered a descendant capable of displaying it as colored letters on a screen, comparing it with a monkey's genome, and returning from that comparison to the bone beneath its own body. Yet even now, the readable archive is much larger than the interpreted one. Human DNA contains remnants of ancient mobile elements, disabled genes, duplications, viral sequences inherited from infections of distant ancestors, and regulatory instructions whose histories are only partly understood. Some traces record events shared with nearly all mammals. Others belong to primates, apes, particular human populations, or single families. Many have no known effect. Some have been recruited into useful biological roles. Others may influence development only under conditions researchers have not reproduced, or may no longer do anything measurable at all. Calling the genome a living fossil should therefore open a question, not close one. A fossil does not have to explain itself to survive, and neither does a sequence. It can remain in the body for millions of years before anyone knows to compare it, before the right species has been sequenced, or before an experiment reveals what changes when it is altered. Under the listener's skin, cells continue copying this mixed archive of active instructions, muted instructions, and historical debris. Among them sits the old machinery capable of beginning a tail, constrained and redirected rather than simply erased. One small insertion helped reveal why that program no longer reaches its ancestral outcome in apes. If a trace that modest can preserve the memory of an entire vanished appendage, how many other events are still traveling inside the body, copied faithfully enough to survive, but unread because no one has yet learned where to look? Press 2 fingers to the base of the spine again. The same blunt point is there beneath the skin, no larger for everything now known about it, no more dramatic in the darkness than it was when this journey began. Yet it no longer feels like a simple bone. Beneath the fingertips lies the reduced terminus of a structure that once extended beyond the body, the anchor of muscles working in the present, and the visible consequence of a molecular change older than the human lineage itself. Now the fingers are resting on several kinds of time at once. One begins only weeks after conception, when a human embryo curves inward and a genuine tail projects from the growing spine. Another begins around 25 million years ago, near the divergence between the hominoid lineage and the tailed Old World monkeys. A third is happening now, as pressure from the chair or mattress reaches the tissues around the coccyx and the pelvic floor adjusts without entering awareness. All three meet under the same patch of skin. The listener does not need to move between them. The bone holds their overlap quietly. Return first to the embryo, because the opening puzzle has changed. The body builds a tail and then reduces it, with programmed cell death contributing to a controlled remodeling that leaves the coccyx behind. At first this looked like an absurd sequence. Construct a feature, supply it with cells and developing vertebrae, then take it down before birth. Why not omit the construction entirely? The answer was hidden in the fact that development does not store a tail as an isolated option, separate from everything else. TBXT is active during some of the earliest and most consequential events in forming the rear of a vertebrate body. It contributes to processes involved in establishing mesoderm, the notochord, and the organization from head toward rear, long before an external tail becomes the feature anyone would notice. Remove its essential activity too bluntly and the result is not simply a human embryo without a temporary tail. Development itself may fail. The old machinery therefore could not be discarded as though evolution were deleting an unnecessary attachment from a finished design. It had to remain available for work the embryo still required. What changed in the hominoid ancestor appears to have been more delicate. An AluY sequence inserted into an intron of TBXT near an older Alu element facing the opposite direction. When the early RNA message folded, the two sequences could pair, bringing distant regions together and encouraging one small exon to be skipped in a hominoid-specific form of the transcript. The gene remained. Much of its message remained. Its vital early roles were not simply erased. Yet the balance among TBXT transcripts changed in a way that evidence suggests contributed to a much shorter caudal outcome. Engineered mice helped make that connection visible, producing shortened or absent tails under related changes rather than proving that one insertion alone explains every detail of ape evolution. This was not a clean switch-labeled tail that moved from on to off. It was a change in how an ancient instruction was read. One output narrowed while older obligations continued. Under the listener's fingers, the coccyx is what a compromise can look like after millions of years. Not the ancestral tail, not complete deletion, but a surviving base around which development could still organize the body. Move outward from the fingertips and the compromise appears across an entire family. Gibbons, orangutans, gorillas, chimpanzees, bonobos, and humans all lack external tails, despite moving through very different worlds. The old proposal that tail loss followed a change in locomotion retains a physical logic, especially if ancestral apes increasingly carried their bodies upright or suspended beneath supports. But the range of living ape movement warns against treating one behavioral story as settled fact. The genetic event can be located more confidently than the ecological pressure that allowed it to spread. Its distribution places it before the ape lines separated, which means no recognizably human ancestor had to lose a tail again. By the time the human and chimpanzee lineages diverged, the absence had already traveled through millions of years of inheritance. Feel the coccyx once more. It is called human because it sits in a human body, not because humans invented it. The history of its reduction belongs first to apes. The later history of how it was incorporated into an upright pelvis belongs more narrowly to the descendants who inherited it. That inheritance was never copied with absolute mechanical uniformity. Human coccyges commonly contain 3 to 5 vertebral segments, revealing variation inside the apparently stable outcome. Rare infants have been described with genuine caudal appendages, although careful diagnosis is needed to distinguish them from other congenital structures and associated spinal conditions. Such cases are individual developmental variations, not a species reversing evolution. Other mammals reach shortened tails by different routes. Manx cats and some dogs carry changes affecting TBXT itself, often altering protein coding sequence more directly than the hominoid Alu arrangement does. The comparison showed neither a universal recipe nor a gene devoted only to tails. It showed a repeatedly sensitive point in a network that builds the rear of the body, where small genetic differences can produce visible caudal changes while also carrying risks elsewhere. Evolution found more than one way to shorten a tail, but none of those ways was free to ignore everything connected to it. Genes are not individual tools stored in separate drawers. They act at different times, in different tissues, among other signals, and one sequence may contribute to outcomes that seem unrelated when viewed only in an adult body. A mutation changes a molecule, and selection encounters the consequences wherever that molecule participates. The tail could be reduced because a change altered the relevant developmental balance without destroying every other task tied to TBXT. What survived was not perfection. It was compatibility. The fingers at the base of the listener's spine are touching that compatibility now. The coccyx receives pressure in some seated positions, allows modest movement in many bodies, and offers attachment to pelvic floor muscles, ligaments, and fibers of the gluteus maximus. A vestige can retain work because vestigial never meant non-existent or necessarily useless. The long appendage disappeared, but its base entered a new network of forces. A structure inherited from one mode of life became a small functional landmark in another, not because evolution anticipated sitting, bipedal posture, or childbirth, but because each new arrangement had to be assembled from anatomy already present. The external tail was silenced as an adult outcome. The remaining bone continued living inside the current economy of the body. Now,